Hello you beautiful people. So video number 4, and today we're going to be covering cardiology. So let's get started. Question 31. Following coronary thrombosis, tissue hypoxia and metabolic waste accumulation during the period of arterial occlusion leads to a transient increase in blood flow. Which term best describes this phenomenon? Is it reactive hyperemia, active hyperemia, functional hyperemia, autoregulation, or reactive hyperemia? So that question just thrown a lot of terms at us. So let's try and break this down a little bit. So coronary thrombosis is also sometimes referred to as a coronary artery thrombosis or a myocardial infarction, and that's the medical term for a heart attack, which I'm certain everyone's heard of a heart attack, unfortunately. So this occurs when a blood clot, and that is a thrombus, forms within one of the coronary arteries, and these are the ones that supply the heart muscle or the myocardium. So this blockage disrupts blood flow to a portion of the heart muscle, and this leads to what we call ischemia, which means reduced blood flow. And if it's not treated, it can potentially cause irreversible damage. So coronary thrombosis typically occurs due to the rupture of what's known as atherosclerotic plaque within a coronary artery. So atherosclerosis is a condition where you have like this buildup of fatty deposits, which we term plaques, within the arterial walls. When a plaque ruptures, it exposes the underlying tissue to the bloodstream, and this triggers the formation of a blood clot, so a thrombus, and that occurs at the site of this rupture. So this thrombus can either partially or completely block the coronary artery, and that's what leads to ischemia or the downstream myocardium. So when we're talking about hyperemia, we're referring to something known as reactive hyperemia, and that is talking about an increase in blood flow to the tissue or organ that occurs following a period of reduced blood flow, this ischemia. In the case of a coronary thrombosis, this leads to arterial occlusion, which causes this tissue hypoxia, which it mentioned in the vignette. The hypoxia means that you don't have enough oxygen going to the area, and in this diagram we can see here that a lot more of it's blue, and then afterwards more of it's red because you have this increased blood flow. So when you have this occlusion, you get hypoxia and you get an accumulation of metabolic waste. It's not being able to be cleared out. When the occlusion is relieved, such as through thrombolysis, or sometimes you just get a spontaneous resolution of the thrombus, the body sometimes is quite good at fixing itself, blood flow to the affected area increases significantly to compensate for the period of reduced flow that just preceded it. And this allows oxygen and nutrients to reach the ischemic tissue, and hopefully this can, make sure if there isn't too much irreversible damage, it can start to clear some of that metabolic waste that's been produced. So this phenomenon is termed reactive hyperemia because the increase in blood flow occurs reactively in response to the previous period of reduced perfusion, rather than it being actively regulated by like local metabolic demands or neural control. So if we go back to our potential answers, the correct answer is reactive hyperemia. And we'll just quickly go through the other ones just so we understand what they are. So active hyperemia is also an increase in blood flow, but it specifically is in response to increased metabolic activity in the tissue. So like if you have like vasodilation to the heart, or like when you're doing exercise, so it's not because of a block, it's because you're doing something that requires more oxygen. So the tissue hypoxia that occurs and the accumulation of vasoactive substances, such as like carbon dioxide, that will lead to vasodilation, and then this is termed active hyperemia. So it's a different terminology when it's that way round. Functional hyperemia is another example, kind of it's of active hyperemia. So functional hyperemia is when there is an increase in blood flow in response to increased metabolic activity in a tissue. So functional hyperemia specifically describes an increased blood flow that accompanies muscle contractions. Okay, so they are very, very similar, almost the same, but active hyperemia is more about metabolic activity in the tissue, whereas functional hyperemia is specifically describing the increased blood flow that accompanies muscle contraction. And I know it's a bit difficult to disengage those, but we don't need to know it more for this question. Autoregulation, so that is the intrinsic mechanism of maintaining constant blood flow to tissue when there is a change in perfusion pressure. So it's a feature of certain vascular beds, especially those in the coronary and cerebral circulation, that tend to be quite reliant on oxygen delivery. So following artery occlusion, there will be an initial reduction in blood flow and perfusion pressure. Local vessel will dilate, and that reduces the vascular resistance, and that restores blood flow despite that reduced perfusion pressure. So this process utilizes a variety of metabolic and endothelial mechanisms, and it's not describing, or isn't the best way of describing, what we see after and occlusion. So it's not autoregulation. And then reactive hyperemia is not actually a mechanism. This isn't real. This isn't a real thing. When you have an increase in blood flow, it's going to be hyper, not hypo. And there is no term of a reactive hyperemia, so it's not that. Okay, so question 32. And again, if there's anything that I haven't explained too well, or any other questions you have, again, please leave a comment and I'll try and get back to it. Okay, so question 32. There are three main groups of muscles: skeletal, smooth, and cardiac. Which of the following describes a unique characteristic of cardiac muscle? Is it that cells have many mitochondria, cells have a centrally located nucleus, cells can contract without neural stimulation, cells are connected by gap junctions, or cells possess a cytoplasmic reticulum and T-tubule system? So yeah, let's just start with basics. Like I said, as we get more of these videos, we won't have to do this. But at the beginning, it's good to come back to our basics. So there are three types of muscle cells in your body, and there are quite a few similarities between them all. So if we go through all of them in turn, in fact, if we go through all of these, I think it'll probably be the best way of doing it. So cells have many mitochondria. Well, let's have a look at this. Yes, cardiac cells do have many mitochondria, but so do skeletal cells. So mitochondria produces ATP, and that's the energy required for muscle contractions. So when you're doing exercise, your skeletal muscles are going to need a lot of energy, and your cardiac muscle, because it's pumping constantly, is also going to need a lot of energy. It's only really smooth muscles that don't have as much mitochondria. They still have mitochondria, but not a lot, which I know is not a very quantitative way of analyzing it. But it would not be this answer, because this is not a unique characteristic of cardiac muscle, because all of your biceps when you're at the gym, your legs when you're running, all of those muscles need a lot of energy as well. So it's not unique to cardiac. So next we have cells have a centrally located nucleus. Okay, well, if we look at the type of cells here and we just kind of look at these structures, and also if you look at the histology, you can see that again, this is not a unique characteristic of cardiac cells. So both cardiac cells and smooth muscle cells have centrally located nucleus. It's only skeletal muscles that don't follow the trend. So skeletal muscles, or muscle fibers, however you want to term it, these have multiple nuclei, and they are located peripherally just beneath the plasma membrane, or the sarcolemma. The reason that skeletal muscles have this, because if you think back to biology, almost all cells will have a centrally located nucleus. Skeletal muscles are quite unique in this. They have it arranged because it allows efficient control of muscle contractions, because the nucleus are positioned closer to where the protein synthesis and other metabolic activities occur within the cell. So skeletal muscles are the odd one out with this. So cells have a centrally located nucleus. That is not unique of cardiac cells, because smooth ones have it as well. So cells can contract without neural stimulation. So due to the presence of pacemaker cells in cardiac cells, myocytes can contract in isolation, and they do not require neural stimulation in order to contract. It's important that the heart is able to contract rhythmically and independently of neural stimulation, because this allows the heart to continue beating even if the connection is severed. And this is where my brain goes. Ever played like Mortal Kombat and you rip someone's heart out, or in even like an old cheesy movie, and they're holding the heart up and you just see it keep beating by itself? That's because of this, the fact that it can pump without neural stimulation. And that's why you get it. Like other parts of your body wouldn't do that. It's just the heart, and that's like very, very iconic in horror movies. So yeah, the correct answer, what makes cardiac cells unique amongst the other ones, is that they can contract without neural stimulation. But we'll just go through the rest of it just so we have a full understanding of this, and I feel like I'm going to say that quite a lot with this video. So cells are connected by gap junctions. So let's actually have a look at what a gap junction is. So both cardiac and muscle cells are connected by gap junctions. So gap junctions consist of these clusters of these transmembrane proteins, and these are called connexins. So these connexins form channels known as connexons, and they span the plasma membrane of neighboring cells. So when connexons from two adjacent cells align, they create this continuous channel, forming what is known as a gap junction. So gap junctions exist to facilitate the rapid exchange of ions, metabolites, signaling molecules, like calcium ions, peptides, small peptides, mind you, in some cases, between like adjacent cells. So this direct communication allows for a coordinated physiological response among groups of cells. And that's why both muscle cells and cardiac cells have them, because both of them, you have a lot of cells altogether that need to work as a group in order for the function to be carried out. So it's important these cells are able to communicate with each other. And then the other thing we're talking about is the sarcoplasmic reticulum and T-tubule system. So here we need to go even deeper. Let me just go forth. So both cardiac and skeletal muscle cells possess a sarcoplasmic reticulum and T-tubules. And so again, this is not a unique characteristic of cardiac cells by itself. We've already ruled out it's not the right answer, right? So the carpoplasmic reticulum. The sarcoplasmic reticulum is a specialized type of smooth endoplasmic reticulum found in muscle cells. So its main function is to store and release calcium ions. And the reason this is important is because calcium ions are crucial for contraction, and we'll definitely be covering that in a lot more detail later on. So when an action potential travels along the T-tubule, It triggers the release of these calcium ions from the sarcoplasmic reticulum into the cytoplasm of the muscle cell. The sudden increase in calcium ion concentration in the cytoplasm initiates muscle contraction by binding to the protein complex troponin. And again, we'll be coming across that a lot more. This is a little bit beyond what we need for this question, but just I'm going to talk about it now when there isn't a question, so that when this question comes up, hopefully you actually think, oh, I've heard of this before at the very least. So it binds to the complex troponin, and that leads to the exposure of the binding site on actin filaments for the myosin head. And that's what allows a muscle contraction to occur. So the T-tubule system is also known as the transverse tubule system. And this consists of these invaginations of the sarcolemma, so that's the cell membrane, and the invaginations are the bits that go inside, and they penetrate deep into the muscle fiber. So T-tubules are essential for transmitting those action potentials from the surface of the muscle cell deep into its interior. They travel along the T-tubule, and they allow for the synchronous activation of the sarcoplasmic reticulum to release calcium ions simultaneously throughout the muscle fiber. So again, it's that thing of all of the muscles need to be acting as one unit. So it's important in both cardiac muscle and skeletal muscles, not so much in smooth muscles. But yeah, this is not a unique characteristic. They are present in both. So the only one that is a unique characteristic of just cardiac muscle cells is that they can contract without neural stimulation. Right. Let's move onwards. Okay. So chronic increase in afterload can lead to pathologic hypertrophy of the heart. What physiological parameter is most closely related to afterload? Is it mean arterial pressure, central venous pressure, end systolic volume, arterial elastance, or end diastolic volume? Okay, and I apologize if this is going a little bit too much in basic. With later videos, I'm just a broken record. Feel free to skip ahead if this is stuff you already know, but if not, it'll be a good refresher. So you have two main systems in the heart. So you have the systole and diastole. So when you take someone's blood pressure, you're going to get two numbers. It'll be like 120/80. So systole is the first larger number, and systole refers to the point at which the heart is contracting, ejecting blood into your aorta and your pulmonary artery. And this is measured in millimeters of mercury, so 120 millimeters of mercury. So the larger number is the higher pressure your vascular system goes to, and the smaller number is the lower pressure the vascular system goes to. So diastole refers to the point during the cardiac cycle when the heart is refilling with blood. This is also the lower end, that 80, so 80 millimeters of mercury. So we have systole and diastole. Now we need to understand preload and afterload. I don't know why the heart looks so angry in this. So preload refers to the volume of blood in the ventricles at the end of this diastolic filling. And it's related to ventricular filling, as it refers to the initial stretching of the cardiac myocytes just prior to contraction. Afterload is the resistance the left ventricle then must overcome to pump blood out. So afterload is measured by estimating the mean arterial pressure. So that gives us our answer. Oh, go green, go green. There we go. Okay, so why is it not the other ones? So why is it not the other one? So central venous pressure. So central venous pressure is the measure of the pressure in the vena cava. So that's the big vein. I mean, you would see it. I mean, it's not exactly an anatomically correct heart, but the vena cava, if we were like looking at it, we would see on this side, and that goes into the heart. So the central venous pressure is a better indicator of preload. And I find knowing this helps a little bit, like it helps with my brain. So, you know, if we take this diagram, the vena cava would be over here, and preload is related to the pressure in the vena cava. The aorta would be over here, and the pressure in the aorta is related to afterload. So preload, vena cava, afterload, aorta, or pressure specifically. So vena cava pressure for preload, aortic pressure for afterload. That's what helps me anyway. Arterial elastance is the next one, and this is defined as the ratio of the left ventricular end systolic pressure to the pressure at the end of the heart pumping out. So this is related to stroke volume. It might affect pressure in the arteries, but the mean arterial pressure is the best indication of afterload, so it wouldn't be arterial elastance. So next we have the end systolic volume, and this is the amount of blood remaining in the ventricle following contraction. So this does depend on both contractility and afterload, and it is an answer that quite a few people go for. So you'd be partially on the right tracks for assuming it, but it's not the best measurement of afterload. And then finally we have end diastolic volume, and this is the amount of blood present in the ventricle prior to contraction. And this is actually a decent indicator of preload, but it doesn't represent afterload. So it is none of these. Okay, question 34. Which valves are open during atrial systole? Is it the AV valves and semilunar valves, the atrioventricular valves only, the mitral valve and the aortic valve, the semilunar valves only, or the aortic valve and pulmonary valve? So let's wrap our head around the four valves, shall we? So there's a few different names that these are called, and you'll find this in anatomy. Like everything seems to have at least two different names for some esoteric reason. But let's kind of go through this. So you have blood, and that flows into the right atrium from both the superior and the inferior vena cava. And this then comes into the right atrium. Blood then passes from the right atrium through the tricuspid valve. Because the tricuspid valve is the one that sits between the atrium and the ventricle, it is known as an atrioventricular valve. Blood then moves up from the right ventricle to the pulmonary artery and passes through the aptly named pulmonary valve. The pulmonary valve is one of the two semilunar valves. These are named semilunar because they supposedly resemble a half moon shape when closed. I personally don't see it, but I'm the kind of guy who says ink splatter at Rorschach tests, so I clearly lack imagination. So blood then goes out, and then when it comes back in, it's a little bit awkward to see here, but when it comes back into the left atrium from the pulmonary vein, it then passes from the left atrium to the left ventricle, and it passes through this mitral valve. This is also the only valve which only has two leaflets. So you can see this, like this one had 3, this one has 2. I mean, that one looks like it has 2 admittedly, but this one is the only one that has 2 leaflets. So it's sometimes referred to as the bicuspid, and this is the second atrioventricular valve. Blood is then ejected from the left ventricle up into the aorta, passing the aortic valve. And this is our second semilunar valve. So the atrioventricular valves are these two. In fact, I'll put that there. So words, English. So semilunar valves are the ones that go from the ventricle into the arteries. So that's the pulmonary and aortic valve. And then atrioventricular valves are the ones that go from the atria to the ventricle, hence atrioventricular. And this is the tricuspid and bicuspid valve. Okay, so now we've got our understand all the different valves. Let's have, well, and actually, yeah, this is what I mean. Like, so three leaflets on the tricuspid valve, 3 on the aorta, 3 on the pulmonary, 2 on the bicuspid. Now, supposedly, these look like half moons. That's why they're called semilunar, and why this one isn't, even though it has 3 leaflets. Again, I don't see it, but I don't go like stargazing very often, so I don't know. But these 2 are the semilunar, and these 2 are the atrioventricular. Okay, so now we've got our head wrapped around the different valves. Ugh. So this is an absolutely horrible graph that you will unfortunately need to become quite familiar with. We're going to be referring back to this pretty frequently whenever we cover cardiology. So don't fret too much if you can't wrap your head around all of it just yet. It's something that you're going to build up over time. So for now, let's just look at systole. So systole is between the first and second heart sounds, these lub-dub noises that you hear. And then if you look at the electrocardiogram, at the start of the systole is where you get that little spike you see in all those medical dramas, the beep, beep you get. So this spike is known as the QRS complex, and that's how the different letters are denoted usually when it's down or up. So QRS. And we get this then afterwards slight elevation to get a QRS. And then this is the ST segment, this section over here. This little bump is the T wave. So QRST. So systole is the point at which the heart is contracting, so that's pushing blood out. And we can see here that the volume of blood in the ventricle goes down until it hits the end. So both the ventricular pressure and the aortic pressure rises until it peaks around the midpoint of systole before then falling again. And this makes sense, you know, we need to increase the pressure to push the blood out of the heart. I mean, a heart is basically just a flesh pump. So with all of that said, let's have another little look at the question. So which valves are open during atrial systole? Now this is something that I think can catch quite a lot of people out, and in all honesty it's because I find a lot of the terminology can get used quite interchangeably. So the question is asking specifically what is open during atrial systole. So if we scroll back down to our graph, what's atrial systole? Well, we can see atrial systole is this little section here, and annoyingly atrial systole occurs during diastole, just to confuse med students further. So whilst diastole is the point at which the blood is filling with blood, it would be incorrect to say it is the point at which the heart is relaxing, because contractions do still occur. So when you think systole, yes, think contraction. However, always be on the lookout in questions like this for any words that come before it. So atrial systole refers to the point at which the atrium is contracting, pushing blood into the ventricles. Ventricular systole is the point at which the ventricles are contracting, pushing blood into the arteries. So whenever you hear someone say systole by itself, they are most likely referring to ventricular systole in actuality, unless it otherwise states. So when blood is being pumped from the atrium to the ventricles, let's go back up here. So when blood is being pumped from the atrium to the ventricles, which valves are going to be open? Well, we obviously need both the mitral and the tricuspid valves open. Otherwise, the blood isn't going to go anywhere. But is the aortic and pulmonary valves going to be open? Well, no, because the ventricles need to fill with blood first before it's injected through. And we can kind of see this a little bit here. So this is where, so the atrial systole is at this point, and you can see here the atrial ventricular valves close at the end of systole, but the semilunar valves will be closed prior to that, and that's where you get this filling of the blood. And you can see here with the ventricular volume, the filling of the blood here, it kind of elevates a little bit, and that wouldn't be possible if the semilunar valves were open. It would just start moving out. So with all of that said, the correct answer is going to be the atrioventricular valves only during this period. Where's the belt? There we go. Let me just cross those out. Okay. Question 35. So, during which phase of the cardiac cycle does the heart receive most of its own blood supply? Is it during diastole, ventricular systole, isovolumetric contraction, atrial systole, or the ejection phase? So, how exactly does the heart supply itself? Okay, so you have these coronary arteries, and these branch off from the aorta. So there might be a bit of a knee-jerk reaction to go, aha, you've just been talking for ages about systole and diastole. Surely it must receive most of its blood supply from the ventricular systole phase, because that's when blood is being pumped out. And you would sadly be wrong. So during the ejection phase, so much force is propelling blood through your aorta that very little of it actually ends up in the coronary arteries. The way I try to think of it is that it's just rushing past so fast that it doesn't have time to go into the blood vessels. However, after the ejection phase during diastole, both the atria and the ventricles relax. I cannot speak today. This relaxation allows the blood to flow into these coronary arteries. So I like to think of it in terms of gravity, and this is not anatomically or physiologically correct way of saying it, but it's what I use to help remember it. So, don't write this on a test paper or anything. So when the heart contracts, blood moves up through the aorta. When it relaxes, the blood starts to pull down because of gravity, and this fills the coronary arteries supplying the myocardium. Again, it doesn't work exactly like that, because if you're upside down, it will still play out. Gravity is obviously not working in that way, but that's just how I remember it for exams, basically. So with that in mind, if we quickly run through the others. So ventricular systole. So ventricular systole involves the simultaneous contraction of both ventricles. This ventricular contraction will actually impede blood flow through the coronary arteries, resulting in less blood flow to the myocardium compared with diastole. So it is not ventricular systole. Isovolumetric contraction. So this is the first stage of ventricular systole. During this phase, the ventricles contract simultaneously, which increases ventricular pressure. However, this ventricular contraction impedes blood flow through the coronary arteries, resulting in less blood flow. Same with ventricular systole. So it's not that one. Arterial systole. So this is the phase of the cardiac cycle which involves both atria contracting simultaneously, as we just saw in the previous question, and this is to force any remaining blood into the ventricles after the usual diastole period. So this atrial contraction impedes blood flow through the coronary arteries, and you're seeing a trend here. These impede it, and it's quite counterintuitive, but once you've wrapped your head around that, it's almost like a lot of medicine is you're learning the exception to the rule, and then as long as you know that the exception, everything else works a specific way, and that's why there's so much emphasis on these exceptions. So again, ejection phase, it's forcing blood through the pulmonary artery and aorta, and that's going to impede the blood flow, so it's not that. So the correct answer to this question is diastole. Okay. Which of the following options correctly identifies the compensation that occurs following a sudden decrease in blood pressure. And I'm not going to read through all of this because my voice is already going. So let's just think through this logically. So when you have a sudden decrease in blood pressure, this is detected by baroreceptors located in the aorta. These baroreceptors send signals up to the brain, which then start compensatory mechanisms to try and counteract the drop in blood pressure. It does this by activating what's known as the sympathetic pathway, the fight or flight response, and decreasing the parasympathetic pathway, the rest and digest pathway. So again, just trying to think, okay, fight or flight, what do I need? What do I need to increase the amount of blood? What do I need to run faster, jump higher, shoot, all that kind of stuff. And with all these, there are three factors here. We're looking at vasodilation or constriction, increase or decrease heart rate, and an increase or decrease of myocardial contraction. And these are all just variations. So let's just go through those three main points. So first of all, is the blood vessel going to constrict or dilate? Okay, so we want to increase our blood pressure, so what do we need to do? So picture this, right? So your dad has asked you to water the grass, and you've got a hose, and you cannot be bothered to walk to the other side of the garden because you're lazy. So you want to shoot that water out further. So what do? You place your thumb partially over the hose, or partially over the opening, and suddenly it starts shooting out with more pressure. And the same as if you're in the shower, if you start to cover up some of where it's coming out, it's going to come out with more force. If blood vessels constrict, the same amount of blood is going to be occupying a smaller space. So the pressure is going to increase. So if we want to bring our blood pressure up, we want vasoconstriction. And then next, what we want to do is increase our cardiac output. So this would normally occur via stimulation of what's known as the beta-1 adrenergic receptors in the heart. So there are two main components of cardiac output. This is inotropy and chronotropy. Inotropy is the force with which your heart contracts, and chronotropy is how regularly your heart contracts. So think ino force, chrono time. So if we want to increase our cardiac output, we want to increase both of these things. So the correct answer to this question is quite simply all of them. All of these will activate to increase our blood pressure. What I seem to find with a question like this and the way it's worded is that the reason that people get it wrong is because they start to second guess themselves. They start thinking, there's got to be a trick to this. Surely there's no way it's just everything. And sometimes there are trick questions, but not all the way. I want to say it's testing your confidence. I sometimes think it's just a bit of an asshole question. And it's not testing your knowledge so much as it's testing your confidence in yourself, which is why you try to believe in yourself, which I know is a silly sentiment to say, but hopefully as we go through more of these, your confidence will grow over time and you'll be able to answer these questions with confidence. Right. Question 37. At which part of the cardiac conduction pathway is there a short delay before the spread of the action potential continues? Is it the sinoatrial node, the right bundle branch, the atrioventricular node, the Purkinje fibers, or the bundle of His? So let's have a quick little look at this pathway, have we? So we have the electrical signal, which starts here at the sinoatrial node. So this is located in the wall of the right atrium. I mean, if you think back to the heart pictures we had before, remember to twist it round because it's a mirror image. So this would be in the wall of the right atrium, and this is responsible for the generation of the initial action potential at the start of each contraction. This signal then travels to the internodal pathways down to the atrioventricular node. So the atrioventricular node is located within the atrioventricular septum, and this acts to delay impulses by approximately 120 milliseconds. The reason for this delay is to allow the atria to fully empty before the ventricular contraction occurs. So there's two nodes. one node initiates and the other delays. So that gives us our answer, but we'll just carry on quickly. So this impulse then travels to the bundle of His, and this is the short section before it divides into the two halves of the heart. And in this diagram, it's called the AV bundle. The bundle of His is an extension of the specialized tissue of the atrioventricular node. However, the short delay occurs at the AVN itself in order to allow the atria to fully empty into the ventricle, so it wouldn't be the bundle of His. This then divides into the left and right bundle branches, so it divides into left and right sides of the heart, which then conduct the action potential to the Purkinje fibers. And these are a network of specialized conductance cells which then carry this action potential back up the ventricles, and this allows the simultaneous contraction of both ventricles. So which part of the cardiac conduction pathway is there a short delay? That is the atrioventricular node. Okay, question 38. Which of the following is the correct definition of ejection fraction? So is it the volume of blood pumped out of the ventricle with each contraction, the percentage of blood pumped out of the ventricle with each contraction, the difference between the end diastolic volume and the end systolic volume of the ventricle with each contraction, the volume of blood pumped out of the ventricle every minute, or the percentage of blood pumped out of both the ventricles and atria with each contraction? Okay, so the fact that this is called a fraction should give you a little bit of a hint here. So ejection fraction is the measure of the heart's efficiency in pumping blood out of the ventricles with each heartbeat. It is calculated as the ratio of the stroke volume to the end diastolic volume. So the volume of blood ejected from the ventricle during systole over the volume of blood in the ventricle at the end of diastole. So this is represented or expressed as a percentage. So ejection fraction is equal to the stroke volume divided by the end diastolic volume times 100%. So let's just quickly go over the other ones so we can wrap our head around this. So the correct answer to this is number 2, the percentage of blood pumped out of the ventricle with each contraction. So let's just go through what the other ones are and know what they're saying. So the volume of blood pumped out of the ventricles with each contraction. This is the stroke volume, and that's why it's involved in our equation, because it's quite closely linked. So ejection fraction is the percentage of blood that is pumped out of the ventricle, while stroke volume is the volume of blood pumped out. Percentage volume. Okay, the difference between the end diastolic volume and the end systolic volume of the ventricles of each contraction. This is just another way of saying stroke volume. So the end diastolic volume is when the chamber is fully filled up, and the end systolic volume is when the chamber has just emptied. So the amount of blood that comes out is the stroke volume. So this is just going to be another way of expressing that same value. The volume of blood pumped out of the ventricle every minute. So this is actually cardiac output. So cardiac output is the volume of blood pumped by the heart per minute. So this to get the cardiac output, it's the stroke volume, so how much blood is being pushed out with each contraction, and then how many times the heart is contracting per minute. You combine both of those together, that will give you the cardiac output. So it's not this one. And then the final one, the percentage of blood pumped out of both the ventricles and the atria with each contraction. As far as I'm aware, there isn't a term for when both the atria and the ventricles contract. So this just, again, if I'm wrong, someone correct me, but I don't think this is a thing that we measure. So just no, it only refers to the ventricles. Right. So question 39, activation of the parasympathetic nervous system decreases blood pressure by which of the following mechanisms? Is it by increasing arterial vascular tone, increasing heart contractility, increasing venous tone, reducing venous tone, or increasing heart rate? So from earlier, we know that sympathetic is the fight or flight and parasympathetic is the rest and digest. So we know that this is going to, in order for the parasympathetic nervous system, is going to decrease our blood pressure. And well, it kind of says that in the vignette, but in case it didn't. However, there is a new term here that we've not come across before. And sadly, I couldn't find a good image to break this down, and my paint skills are atrocious. So we're just going to have this little visual aid to cheer you on for getting this far. Okay, so let's break this down a little bit. Venous tone. So venous tone refers to the degree of constriction or dilation actually, which occurs in the walls of the veins. So this is going to affect the veins' diameter and compliance. When veins constrict, that is an increase in venous tone. So their diameter decreases and they become less compliant. This constriction increases the pressure within the veins, reducing their capacity to store blood. And as a result, more blood is returned to the heart, leading to increased preload. Increased preload can enhance stroke volume, cardiac output, all of that. So that's going to then increase blood pressure. Conversely, when veins dilate, so a decrease in venous tone, their diameter increases and they become more compliant. This dilation reduces the pressure within the veins, allowing them to store more blood. So less blood is returned to the heart, and that will reduce preload. So decreased preload can decrease stroke volume and cardiac output, and then this is going to decrease blood pressure. So all of that was just a quick overview of what venous tone is, but in reality, if you look at the answers here, you'll notice a trend. So 4 of these are going to be activated by the sympathetic pathway. Incidentally, 4 of these also say increase. Really, even if you forgot which way round parasympathetic and sympathetic was, just pick out the one that is the odd one out, because you can kind of tell that all of these are going to increase blood pressure, only one of them is going to decrease it. So a reduction in venous tone is going to decrease blood pressure, and all the rest of these would have the opposite effect. Arterial vascular tone, heart contractility, venous tone, heart rate, all of those would increase the blood pressure. Okay, so last question. Thank you for sticking me with this one. Okay, in a non-pacemaker cardiac cell, which ion channels trigger depolarization? So is that ligand-gated sodium channel, sodium-potassium pump, voltage-gated calcium channels, voltage-gated sodium channels and voltage-gated potassium channels, or ligand-gated calcium channels? So in terms of cardiology, this is the other horrible diagram that you will need to become intimately familiar with. So, and also just, it starts at phase 0 rather than 1, just to be annoying. Scientists, am I right? Okay, phase 0. So this is rapid depolarization. This phase is initiated by the influx of sodium ions, and this occurs through fast voltage-gated sodium channels, which causes this rapid depolarization of the cell membrane. This rapid depolarization leads to the upstroke of the action potential. Now, while it doesn't show it on this diagram, and in all honesty, frustratingly, it doesn't in most textbooks, potassium also plays a role in this section. So in non-pacemaker cardiac cells, a small number of voltage-gated potassium channels may open during the initial depolarization, and this contributes to a small efflux of potassium ions. This efflux, this efflux of potassium ions, helps to counterbalance the sodium influx. And this helps to contribute to the initial repolarization and termination of the depolarization phase. The role they play is small, so a lot of textbooks do only refer to sodium, but it's important to remember that potassium is slightly implicated in this phase. And then we go to phase 1, so this is initial repolarization. So afterwards there is a transient outward movement of potassium ions, and then the inactivation of sodium channels, resulting in a brief repolarization. That's what we have up here, transient outward. Phase 2 is the plateau phase. So during this phase there is a balance between the inward movement of calcium ions, and this occurs through what are known as L-type calcium channels, and then the outward movement of potassium ions. So the balance between these two ions leads to this plateau phase in the action potential, and this helps to sustain muscle contractions and helps prevent premature repolarization. Then we get to stage 3, which is the repolarization. So here there is an efflux of potassium ions, and that leads to repolarization of the cell membrane and restoration of the resting membrane potential. And then finally you get to part 4, which is on both ends of this, and this is the resting membrane potential. So what is it, the normal state? So after you've had the repolarization, cell returns to its resting membrane potential, and it's ready for the next action potential if it's needed. So in non-pacemaker cells, so ventricular cells, this action is produced predominantly by the activation of sodium channels, this depolarization phase, and that increases the membrane conductance flow of sodium. These channels are activated when an action potential arrives from a neighboring cell through the gap junctions, what we looked at a little bit earlier. And when this happens, the voltage within the cell increases slightly. If this increased voltage reaches a certain threshold, which is about -70 MV, it causes the sodium channels to open. And this produces a larger influx of sodium into the cells, rapidly increasing the voltage further to around +50 MV towards the sodium equilibrium potential. So with all of that said, the only answer that would make any sense here is voltage-gated sodium channels and voltage-gated potassium channels. So why isn't it ligand-gated ion channels? So ligand-gated ion channels are only open when they are bound by a neurotransmitter to change the resting potential of a neuron and trigger an action potential. They are not opened by the depolarization phase of an action potential, but instead trigger a post-synaptic potential. So when you're referring to the cardiac, and when you're referring to this initial action potential, the depolarization phase, think voltage gated, not ligand gated. Sodium potassium pump. So these function during the repolarization phase. So around this phase. And these are responsible for they remove 3 sodium molecules out of the cell in exchange for 2 potassium molecules. So that's not implicated in the depolarization phase. Voltage-gated calcium channels, they are not in the depolarization phase. So you can see they are implicated during the plateau phase, not during that. So calcium are implicated more during phase 2, not phase 0. And then finally, ligand-gated calcium channels. These, again, think ligand with the sodium channels. So they only affect when they're bound by a neurotransmitter and are involved in post-synaptic potentials. This is not to do with depolarization. So That's it. That's the 10 questions for today. Thank you very much for sticking with me. My voice is starting to go. I probably should have stayed off a little bit. But thank you for sticking with me, and good luck with your studies. All the best.